क्या वीडियो किसका वीडियो भेजता है तू?
अरे ऐसे ही रखता हूँ क्या मैं आरोपी भेजा है
क्या है
मेरा फोन है ये कौन सा रास्ता चल रहा है मेरे एक चैप्टर में चल रहा है इसकी कॉपी देखते हैं ये टाइम है कौन सा रास्ता
Good evening. What was it? Circulatory system you were studying, no? Yes. Nobody brought the book. Very good. Good evening. Yeah. She's sitting for. Is it remote? Okay, the other day we were talking about blood cells. Let us continue from there. We were talking about the structure of the RBC. The other day we studied about RBCs. Did we finish structure of the RBCs? Yes. We finished all, no? So what are some of the important properties you have to remember? RBC, the structure. Put one off. On. Both are on. Put one off and the other temperature. See what's the temperature? Yes, ma'am. For this one. Put it at 24. No. 24. Yes, ma'am. So now, when we are talking about what are the properties about RBCs you have studied? Shape is biconcave because of the absence of the nucleus. And instead of the nucleus, what is there? Hemoglobin is there. Hemoglobin is a respiratory pigment we have done. So I think those compounds we haven't studied. We should be about RBCs. Did I move on to WBC already? Yes. Compounds of hemoglobin, did we study? Yes or no? I don't think so. So, no, that is different. When I talk about hemoglobin, just check if I have discussed this in case we miss out, because this is important for you. Hemoglobin, I must have told you that hemoglobin has got two components, no? Heme or heme and globin. This we discussed, no? Yes. And then when I talk about this hemoglobin has got affinity with respiratory gases. Now, what are the three respiratory gases it binds with, and what type of compounds does it give rise to? So this is very important for you to understand. Let us start discussing it this way, that if I talk about hemoglobin and oxygen, what is the compound form? Oxyhemoglobin. Oxyhemoglobin. What about hemoglobin and carbon dioxide? Carb-aminohemoglobin. Hemoglobin, yes. And carbon monoxide, carboxyhemoglobin. Carboxyhemoglobin. When I talk about the difference or comparison between these three compounds, when we are talking about carbon monoxide, I must have mentioned that carbon monoxide has got a greatest affinity of combining with hemoglobin, or say the other way, hemoglobin combines more readily with carbon monoxide in comparison with oxygen and carbon dioxide. So if I understand this way, that once this compound is formed in the body, what will happen? This makes the blood venous. The blood becomes venous, and this condition is known as carbon monoxide poisoning. This is what is known as carbon monoxide poisoning. When will this happen? When is the chance that this may happen in our body? When we are in an extremely polluted area. When we are in an extremely polluted area, this condition may happen where we come across that the affinity, since the affinity of hemoglobin is more for carbon monoxide and the binding of hemoglobin with carbon monoxide has happened. So the oxygen binding capacity decreases. The oxygen of the atmosphere, even if it enters our lungs, it cannot bind with the hemoglobin present in our blood. So in that case, what will happen? We'll start suffering from breathlessness. So that's the reason whenever you are in a suffocated condition, ill ventilated room, or in a condition where it is too smoky, it's too polluted, what happens? We suffer from breathlessness, and there is a discomfort that we come across. Why that? Because the tissues are not getting adequate amount of oxygen at that time, because the oxygen binding capacity starts getting decreased. So that is what it is there when we are talking about the venous condition of the blood. So try to understand what is meant by venous blood. So about the number we must have discussed. No? What about the number? And then the site of origin. This is also something you have to remember for questions. Origin, when I talk about where is the blood, where is the RBC originating? What did I tell you? Red bone marrow, if it is for the adult. And if I talk about for a child, growing children, apart from the bone marrow, it can also be growing in the liver and the spleen. In the liver and the spleen also. But if I'm talking about the embryonic condition where the liver and the spleen and the bone marrow, nothing is getting formed, at that time we can understand that liver also basically can be, once the liver is formed, that can be a site for the formation. Why is, I think these things I must have discussed already, the spleen, why is it known as the graveyard? And at the same time, what happens to RBC in the liver? That is integration of the hemoglobin, that part we already discussed. Then what are those terms that we have come across? What are those terms? Erythropenia and polycythemia, abnormal increase and abnormal decrease to understand. So next let us talk about leukocytes. So now for leukocytes, what did we understand? Leukocytes, why are they termed as such? Because they are colorless. And what are the two categories we studied? The two categories that we studied are granulocytes and agranulocytes. No? Granulocytes and agranulocytes. So when we are talking about agranulocytes and granulocytes, what would be the basic difference between them? The basic difference between them would be just the presence of some substances in their cytoplasm. If I talk about granulocytes, some additional granular substances, majorly proteins, are present in the cytoplasm. And if I talk about agranulocytes, the cytoplasmic regions are generally, firstly, the cytoplasmic region is very, very scanty, very small amount of cytoplasm, little amount of cytoplasm is there. And on top of that, we don't come across this type of granular substances at all in the cytoplasm. So this is what we are trying to understand, a major difference between granulocytes and agranulocytes. But what are the types if you now try to understand? Granulocytes, what is the first type of granulocyte? Neutrophils. 5 types, no, we must have already discussed it other day. 5 types, and then next is I talk about the eosinophils, and then I talk about the basophils. So 3 types of cells. So what are the 3 types of cells? Neutrophils, basophils, eosinophils. If I have to talk about the granulocytes. And what about the agranulocytes then? The agranulocytes, one is monocytes, and the next is lymphocytes, monocytes and lymphocytes. Now, let us try to understand some special properties of all these cells. Just trying to understand that when I talk about generally origin, what are I supposed to understand? Is this common in all of the cells? More or less common. When we are going to come across that the origin is going to be bone marrow. Number also we discussed that. What was the number that we discussed? What was the number? 4,000 to 9,000 cells per cubic millimeter of blood that we already discussed. No? Now, when I talk about what is a common feature for all these, if I have to talk about granulocytes, they have lobed nucleus. That's a common feature. And what about this one? They have large or small nucleus. So when I'm talking about the nuclear region, see here, if I talk about the lymphocytes, the nuclear regions are comparatively small as compared to monocytes. Monocytes majorly would be a cell, something also drawn this way, where the nucleus is almost covering up the entire region of the cell, leaving a small rim of the cytoplasm. So all these diagrams that you come across, either drawn this way or this way, all are monocytes. And then lymphocytes comparatively can be 2 types. Either this way you get a cell that is also lymphocyte, or if I talk about this type of a cell, this is also lymphocyte. But majorly when you talk about the monocyte, the nucleus is roughly bean-shaped nucleus, but a big nucleus it is. But what about the neutrophil, eosinophil, basophil? Try to find out the major differences you are asked to identify the figures. Neutrophils majorly 3-lobed. Eosinophils majorly 2-lobed and 2 to 3-lobed. And basophils, they are actually indistinctly lobed. They also have a nucleus. But the mass of nucleus and the distinct lobes will generally cannot understand as clearly as compared to the other two. What are some of the special properties of these three that we have to talk about? Neutrophils, one thing very, very clear to understand if I'm talking about neutrophils, they are the maximum type of cells. Neutrophils are the maximum number of cells that we come across in the total count of WBCs. In any amount of the blood, whatever amount of WBCs would be there, maximally they would be all neutrophils. Maximally they would be neutrophils. What about the other properties of neutrophils? When I'm talking about neutrophils, they basically undergo the process called phagocytosis. Do you remember what is phagocytosis? Engulfing. So last class did I talk about the amoeboid feature and the concept of diapetesis?
Yes, ma'am.
What was diapetesis? I told you.
Losing out of WBC from the capillaries
For
phagocytosis.
to clear an infection. So now that I'm talking about what is diapetesis, the squeezing out of the WBCs through the capillary wall to reach the site of infection. And after they reach the site of infection, how are they going to treat? How are they going to treat the germs or the pathogens? By engulfing them, by completely taking them inside and digesting them. So if that, put this off, put this fan off. I'm not going to go. So now that when we are talking about that, what is phagocytosis or what is the method of defense for the WBC? WBCs can show two methods of defense. One that we are talking about is phagocytosis. So neutrophils undergo the method of phagocytosis. What about eosinophils? One property, okay, one more thing that we have to understand for this type of cells is what type of dyes we can generally use for them. Now, what is the meaning of dyes here? Dyes means I make a preparation of the blood smear. No, how is it made? We take a glass slide. The glass slides we take. In the laboratory, sometime or the other must have seen that. The glass slide, put a drop of blood, and then squash it, and then dry it, and then put alcohol and the stain. And now that you put the stain, I want to see neutrophils. Then what type of stain I should use? Dye I should use. The dye should be a neutral dye. What is the meaning? Now see, most of the times whenever we are talking about this coloring substances, the dyes and the pigments, either they can be acidic or neutral or basic. So here I want to see the neutrophils only very clearly in the smear. Then I have to use a neutral dye. Or if I say the other way, I am using a neutral dye, then which type of WBCs I will be able to see predominantly in the slide? Neutrophils. Because this type of dye will give a color to the nucleus of neutrophils. Then what do you think? Now that I talk about the eosinophils, then what type of dye should be used? For eosinophils, basically the acidic dyes are used. For eosinophils, basically acidic dyes are used. Check in your table, there should be a table there. Acidic dye should be used. If I talk about eosinophils, acidic dyes. No? Correct. Yes, acidic dyes are used. So when we are talking about this, acidic dyes are used. So that means if I use any acidic dyes, then only this type of cells I will be able to see. But what is their method of defense? They are engaged in releasing specific type of chemical substances known as antitoxins. But occasionally they may engulf germs, but they are not basically engaged in phagocytosis. Try to understand, now that these 5 types of WBCs we are talking about, either they undergo phagocytosis or they release chemicals. These are the 2 methods by which they give us defense. Either by phagocytosis or by the process of, or by releasing chemicals. So now antitoxin, of course, is a type of a chemical that we are talking about now. Okay. What about basophils? What type of dye? Basic dyes. Basic dyes. And what is their method of defense? Their method of defense is they would be releasing special chemical substances like heparin. Okay, they would be releasing heparin. I think that should be there. Heparin and histamine. Correct. Heparin and histamine. So there's any of these 2 you can remember. Heparin or histamine. Both are the same things. So when we are talking about heparin or histamine, these are the chemical type of substances which are released from this. So what did we come across in all these three? The type of cells which are maximally present, this point you should always remember, neutrophils are the type of cells maximally present. And how do they bring about defense? If I talk about these two, they release chemicals. And if I talk about this, they undergo phagocytosis. Come to this category now. Monocytes, lymphocytes. Again, when I talk about monocytes, here to understand, if I'm talking about monocytes, these type of cells are again engaged in the process of phagocytosis. A big bean-shaped cell, a nucleus, scanty cytoplasm, engaged in the process of phagocytosis. Comparatively, if I now talk about lymphocytes, they can be small lymphocytes or large lymphocytes. Actually, there are specific categories known as T and B lymphocytes, so we don't understand that right now. How do they show defense? They show defense by releasing special chemical substances known as antibodies. This is very, very important for you. It comes in your paper. Antibodies. What type of chemical substances do they release? Antibodies. Antibodies are released by lymphocytes only. So now that we are talking about the special properties of this and the figures to remember. This figure. Sometimes you are asked to draw also diagrams. This cells, you should know how to roughly draw the diagram. Draw the structure of the lymphocyte and certain questions, a full 5-mark question. That type of a question also you need to prepare it. So this is about the cell types. What is about WBCs? You have to remember. Did we talk about leukemia and leukopenia? Did we talk about that the other day? Leukemia and leukopenia. So if I have to talk about abnormal count, so these two terms have to be understood. Leukemia and leukopenia. The difference that you have to remember. So for WBCs, this is what you have to understand. Take a note of these terms. Again, I write leukemia and leukopenia. What is their difference? You are asked in the difference section also. Differentiate between. These are terms where you get questions on. Leukemia, erythropenia. What is the difference? The third type of cell that we have to talk about would be platelets. Now we talk about platelets. The third type of cells we look into would be platelets. What is the other term for platelets? Thrombocytes. Now, this type of cells, thrombocytes or platelets, they are unorganized cells. Unlike the WBC and the RBC that we talk about, these are not well organized cells. If I show you again the structure of a platelet, the platelet more or less appears this way. Fragments as if they are, no cell fragments as if they are. So these are unorganized cells. And they also don't have nucleus. The platelets, again, if you have to try understanding some features, they also don't have nucleus. They are also enucleated. No nucleus. Then, what are some special properties of the platelets again? When we talk about platelets, where do we come across platelets? Where exactly we come across the major accumulation of the platelets always at the site of injury. Always at the site of injury. What about their site of origin again? If I have to talk about, remains more or less the same. Here also you have to remember this as bone marrow. About the platelets more. What about their number? What about the number of the platelets? It is basically 2.22 to 4 lakhs, no? Per cubic millimeter of blood. 2 to 4 lakh cells per cubic millimeter of blood. It can be 2.5 to 4.5 also written in your book. Roughly that number we understand. 2 to 4 lakh cells per cubic millimeter of blood. यहाँ पे function सबसे important है that you have to remember. What is the function of this type of cells? Function of this type of cells is helps in the process of clotting. What is the meaning of the process of, what is clotting? When should clotting be done? What is the difference between the word clotting and the hemorrhage that we understand? The difference between these two terms is if the process of bleeding is happening inside the body because of some injury and the clotting does not happen inside the body, actually the bleeding should not happen inside, internally. If that happens, hemorrhage, and if the process of clotting does not happen, that's a major problem. Otherwise, basically we associate the word clotting with some external injury. So suppose we suffer from some cut, wound, fall, etc. At that time, when the skin is bruised and the muscles are torn, what happens? The process of bleeding. And normally, within what time frame the process of the oozing of the blood should stop? It should stop maximally within few minutes. If that does not happen, then it is understood that that must be because of the low count of the platelets or a massive deficiency of vitamin K or calcium, any of these three factors. No? So here right now we are talking about clotting. So what is clotting? Clotting is a process of closure of the or stoppage of the process of oozing of the blood due to the reactions carried out by the platelets. Can we talk about the abnormal count here? Yes, here also we talk about thrombocytopenia and I think for Selena, just check if these terms are there. I don't think Selena, it must not be there. Thrombocytopenia, thrombocytosis, thrombocytopenia. So when we are talking about this, when we are looking into this is optional for you since we are talking about this here. Now we move on to this concept. Now after these three types of cells that you are aware about, platelets, and then we talk about the RBCs and the WBCs. Here, related to this, their specific function, site of origin, everything. Here let us now talk about, since we are talking about injury and healing and clotting, let us also talk about one word known as serum. What is serum? What is serum? Serum would be, this definition is very, very important for you. What is serum? Serum is the plasma without the fibrinogen. Now, why fibrinogen has to be removed out? Take a note of this definition. What is serum? Serum is the plasma from where the protein fibrinogen is removed out. The process from where, the plasma from where protein fibrinogen is removed. The protein from where, the plasma from where, the plasma from where protein fibrinogen is removed out. But how fibrinogen is removed out? Because fibrinogen is always used in the process of clotting. First of all, write down the definition, serum. Serum, what is the definition? Plasma from which protein fibrinogen is removed. So always try to understand, now that I'm talking about clotting, now that I'm talking about a wound healing, injury healing, did you ever notice that when the clot is young enough, and by chance if you just happen to pull that or strip that clot or that brown skin which is getting formed, by chance if you try to strip it out, peel it out, did you ever notice a light yellowish colored fluid oozes out from the site of the wound? That is what is called the serum. Once the clot is fully formed, say suppose there's an injury, the clot is fully formed, then it appears as a black patch over the skin. It's a black patch. But before the clot is done completely, it's not yet dried. If it is fully dried, the black skin falls and that region becomes a little bit whitish. But it is not black yet. It is still light brownish. It is still light brownish. Then, at that time, suddenly if it gets torn or if you happen to peel, you get to see a light yellowish color fluid oozing out. That is what is the serum. So, actually, what is the composition of the fluid? The composition of that fluid is actually plasma. Fibrinogen already used up because fibrinogen is a major protein used in the process of clotting. So, plasma fluid that we talk about without fibrinogen is the serum now to understand. So next what we are going to move on to is, we try to first of all understand what are the steps of blood clotting. Since we are talking about platelets, what are the process of clotting we talk about, what are the steps of blood clotting? Let us try to understand that. That's very, very important. Steps of blood clotting. What is blood clotting? When does blood clotting happen and what are the steps? This particular flow chart that we are going to write now is very, very important, and you get this as it is for 3 marks in your paper. You get this as it is for 3 marks in your paper. So now, when we are talking about blood clotting, under what different conditions that happen? Which type of cells are important? So here, first of all, to understand is at the site of injury, which type of cells will majorly try to play a role? The type of cells are platelets, and they generally have a capacity of releasing certain enzymes known as thrombokinase. They have a capacity of releasing an enzyme known as thrombokinase, also known as thromboplastin. Thrombokinase, also known as thromboplastin. So where is this release happening? When the platelets are inside the bloodstream, are they releasing this enzyme? No. When the bloodstream is torn and there's an injury, at that time the platelets start releasing this enzyme. Now that we are talking about this thromboplastin or the thrombokinase, this particular enzyme, first of all, if I show you this, that whenever there's, you're not able to see that side? Not able to see? When we are talking about that there's a site of injury, the blood vessels are torn, at that time these reactions will happen here. So what is the first? The first step is the release of the enzymes one after the other, but in a cascade. We are going to learn this sequence in a cascade, meaning the release of one component will trigger the formation of the second, and the second will trigger the formation of the third. It is that way. So first of all, when we are talking about that there are a large number of factors here. So what will be the sequence? First of all, we are talking about this thrombokinase. Of course, we understand that these different type of proteins and pigments are always released in the inactive form. But we can start from this step also. But here I show you this way that this thromboplastin also is released first of all by the platelets in an inactive form. Why the word pro here? It's a precursor, which means that first of all the platelets releases them in their inactive form. Then this, in the presence of calcium, becomes active. Calcium where? In the blood, of course, becoming active. Thromboplastin. This thromboplastin is majorly now present in the site of injury. Now the thromboplastin is there. Another particular protein, prothrombin, now starts. Don't write this. We will write it exactly the way you are asked in the exam. So this thromboplastin, these are just the steps first to understand. This thromboplastin, once it is in the blood, then this thromboplastin will convert prothrombin to thrombin. Now what is thrombin? Prothrombin, another type of a plasma protein, that is. Another type of a plasma protein. Now the thrombin is present. This thrombin will now convert fibrinogen. You already know fibrinogen, a plasma protein. This fibrinogen will convert into fibrin. Now that the fibrin is present at this site of injury, now what will happen? The fibrin is now present at this site of injury. The RBCs start clumping together. The fibrin protein is there. And this fibrin protein initially, which was getting soluble in the plasma, now it starts getting converted into some thread-like structures, a meshwork, where all the dead RBCs are trapped. Where all the red RBCs are trapped there, the dead RBCs are trapped there, or the RBCs die out at this exposed region, and resulting in the formation of that brown colored skin that develops here, which is known as a soft clot. So that fine threads of fibrin and the RBCs result in the formation of a nodule-like structure here, blocking the entire the release of the blood, the seeping out of the blood, and now the soft clot is formed, which keeps on drying out, accumulation of the RBCs, which keeps on drying out, And later, it results in the formation of the hard clot, which we call it to be almost dark in color. At this site, two things may happen when we are talking about the pus formation. What is pus formation? Quite a number of times, the dead WBCs start accumulating in large numbers, resulting in the formation of a whitish creamy substance, which may start oozing out from the site of clotting. That is called pus formation, remember. But if I talk about serum, already you can differentiate. Now, if the clot is not fully formed and if it is torn, then the serum will ooze out. Otherwise, the serum will not ooze out. How are you now supposed to write down this? Let us write it down properly. All of you. Platelets at the site of injury. Platelets at the site of injury. At the site of injury, what are they releasing? They are releasing thromboplastin or thrombokinase. They are releasing thrombokinase or thromboplastin. Then this thrombokinase or thromboplastin is converting prothrombin to thrombin. And this step will take place. This step will also require the presence of calcium ions. This will also require the presence of calcium ions. Always to remember, with reference to enzymes, also last year you have studied, whenever you are talking about any compound which that's a precursor form, inactive form that is. Now, this presence of thrombin will also result in the formation of fibrinogen or conversion of fibrinogen to fibrin. This is the soluble form and this is the insoluble form. Plus, with this fibrin, now RBCs or dead RBCs, resulting in the formation of soft clot. And this results later in the formation of hard clot. Okay, so this way, exactly this way, you are supposed to write it. And this entire flow chart comes for 3 marks also to be written in your paper. Exactly this way, step by step, you are supposed to write. This is called a cascade of reactions. Cascade, why? Because one compound activates the formation of the other. Based on this, diagram-based questions also you generally get. Next is we try to come across the role of, okay, we were talking about vitamin K, the role of vitamin K and calcium. The role of vitamin K and calcium. Here also calcium is actually necessary, vitamin K and calcium. Now, sometimes you are asked in your paper also, name the mineral element which helps in the process of clotting. Mineral element, calcium. Name the vitamin which helps in the process of clotting, vitamin K, to be written that way. So this is the concept of clotting to understand. Now, here we talk about the concept or disorder, rather, we are talking about hemophilia. What is hemophilia? Hemophilia is a genetic disorder. Already we have studied genetics, no? Yeah? I think there also we must have talked about hemophilia, where now that we are talking with reference to lesser number of platelets, and we are saying that if there's a deficiency of platelets, the person may suffer from hemophilia. Okay. But actually, the lesser number of platelets is because of some genetic disorder. What is the genetic disorder? And what is the cause of the genetic disorder? This person who is suffering from hemophilia has a recessive gene in the X chromosome. has a recessive gene in the X chromosome, and it is only because of the presence of the recessive gene the individual is suffering from hemophilia. So, what are the symptoms of hemophilia? From the smallest cut and wound, also the bleeding will be profuse, and the process of clotting will not happen regularly. It will take a very long period of time, unless and until certain coagulants are given to that person. Something like heparin. Heparin is released inside our body naturally, so that inside, if there is some hemorrhage, there should not be any clottings. As I said, that when we are talking about these anticoagulants like heparin and all, they actually do not allow the process of clotting. So similar type of chemical substances are there, which will generally prevent the process of clotting inside the body. They can be given in the form of injections also. So here, hemophilia is a condition where we are saying what can be some of the root cause. Of course, it's a genetic disorder, but deficiency of platelets can also result in this disorder. Profuse bleeding is one of the most common symptoms, even from the gums. The bleeding can be profuse, even to death. So that these are some of the conditions that we understand. Can it be? Is it curable? Generally not. It is not generally curable. So these are some basic concepts about hemophilia that you have to understand. Next is profusion. Now let us talk about the concept of blood groups and transfusion. What is the meaning of blood transfusion? What is the meaning of blood transfusion? Why is blood transfusion done? Transfusion suna hai na, kabhi to koi suna hoga. What is the meaning of blood transfusion?
If a person has blood cancer,
Acha.
then we have to replace that person's blood.
That is replacing altogether. But transfusion of blood meaning many times, even if a person is going through minor surgeries also. Say suppose a person has suffered from a massive accident and then so much of blood is lost from the body. Then what is happening? The patient is almost suffering from massive loss of blood. Then even if the adequate amount of blood units has to be given to that person. So what I said, a patient or a person who has suffered from a major blood loss due to any reason, accident, surgery, etc., or organ transplant, ऐसे वाले situation में the person may be in need of blood. So, can anybody's blood be just given to that person? No, there has to be a lot of tests and a lot of checking. No? So, what are they actually checking for? What are they trying to find out? They are actually trying to look for certain antibody and antigen reactions. So, in this entire concept of transfusion, so what is the meaning of the word transfusion basically? Transfusion means mixing of the blood. Mixing of the blood, where there has to be a donor and there has to be a recipient. So now I talk about the person who has some basic blood loss. So it is actually the exchange of the blood between two individuals, where we are talking about it can be either a person who is suffering from a disease, it can be a person who is suffering from who has gone through a surgery, accident, etc. So between the donor and the recipient, there is an exchange of blood. But while this blood has been given, raised to the body of the recipient, now the donor wants to give the blood. So what are some of the factors that has to be checked here? The factors that has to be checked here are many. Where certain antibodies present or not, that has to be checked. Certain antigens present or not, that has to be checked, and many more. Right now we are talking about this too. Antigens and antibody. What is the meaning of this? Antibody just now you have come across, no? What are antibodies? Certain proteins that the blood cells itself release. Antibodies, where are they present? Just now you have studied. WBC is releasing antibodies. So these are proteins which have already dissolved in the plasma. So the WBC are releasing those antibodies which are proteins. And I would say, try to differentiate this way. These are the proteins. Antibodies are proteins released by what? Released by WBCs. And where are they present? They are dissolved in the plasma. So that's the basic concept of antibodies. Now you know. Then what are antigens? These are also proteins. But these antigens, our body actually is not producing, and it is entering our body from outside sources. Any protein of a foreign substance by the way of our blood, by the way of infection, etc., which is entering our body, all can be categorized as antigens. So I would say this way, then what are antigens? Antigens are actually foreign proteins. What is the meaning of foreign protein? Not of our body's own. So here, these two things are to be taken care of. That this protein inside our body and this protein, are they directly entering into a massive reaction and resulting in clotting inside the body? If that is happening, then that's dangerous. So during this process of transfusion, that the donor is giving blood to the recipient, this reaction between these two types of proteins should not happen. If that is okay, then no problem. The donor can give blood to the recipient. Try to understand, we'll be now talking about these two types of proteins. So what I said, two types of proteins are to be taken care of. One type of protein that our plasma already has, which the WBC is producing, and another type of protein which are entering from outside. Now again, this protein which are entering from outside, I now can also say that one example of such type of proteins are also the proteins on the RBC surface. Also can be proteins on the RBC surface. Let us see how. In this case, now I talk about, based on this concept, we are going to now understand what are blood groups. Blood groups, based on this presence of all these proteins, etc. We can differentiate groups, or we can differentiate the blood type that we have into four main types. The blood types that we have into four main types. What are those blood types? I talk about blood group A, or I talk about blood group A, and then I talk about blood group B, and then I talk about blood group AB. Do you all know your blood groups?
Yes.
Yeah.
Okay. And then I talk about blood group O. Human can be categorized based on these four blood groups. Okay. So now if I'm talking about blood group A, what are the two things I have to understand? The type of protein that is there in the plasma and the type of protein that is there on the RBC surface. On the surface of the RBC. So I'm trying to understand the type of protein on the plasma and the type of protein on the RBC surface. So if I talk about people who are who have blood group A, they have in the plasma antibody A and antigen A. Antigen. What is that word? Antigen. Antigens are also a category of proteins. And antibody is also a category of proteins. Simple, try to understand what I'm saying. A person who is having blood group A, what type of proteins do they have? A type of protein on the plasma that is known as antibody A. Antibody we generally write with small letters. Always remember, antibody we generally write with small letters. And antigens we would basically write with capital letters. Here they have written capital. Majorly we write antibody with small letters. Antigen with capital letters. Okay? Now we talk about B. So you should be understanding. If I'm talking about B, then this person should have antibody B in the plasma. It is by the WBC. And what about this? Antigen B on the surface of the RBC. Okay? आ रहा है समझ में? A person with blood group AB. बोलो.
Antibody उल्टा होना चाहिए.
Antibody.
It should be soft.
Antibody.
Antibody should be soft, right?
Antibody B, correct. Antibody B I said, no? Antibody B and antibody A, correct. So I said antibody B for people with blood group A, correct? Antibody B with people of antibody A for AB, that's correct. Now I talk about people with AB. AB में कोई भी antibody नहीं होगा, and they will have antigen A also and B also. What about group O? They will have both anti-A and B, and they will not have any antigens. Now, just these four things you try to understand first. अगर ये clear है, then the process of transfusion would be very easy with the logical explanation, because you are getting questions that way. So first of all, if I try to understand, now say I try to understand a person who has this antibody and this antigen, will the process of clumping happen? Process of coagulation and clumping के लिए this type of antigen and this type of antibody may result in clumping. Maximum chances हैं. So in our body it is already said that way that a person who is having blood group A has got this antibody and just the other type of antigen. So clumping इसीलिए नहीं होता है. What is the other word for clumping? Get acquainted with that word? Agglutination. Agglutination. What is the meaning of agglutination? Agglutination means clumping. Agglutination. Or clumping. So that means when these two come together, there may be chances of coagulation, clumping. When these two come together, there may be chances of coagulation or clumping. Now, I say that these type of people who are having blood group AB are known as universal recipients. These are called universal donors. Let us see why. When we are talking about people having blood group O, they are called universal donors. Now why? This person can give blood to this also. Yes? This person can give blood to this also. Try to understand the logic. Why? ऐसे क्यों? Problem क्यों नहीं होता? Because they have got only small amount of that antibody. Antibody when enters into this person, plasma में तो वैसे भी कोई antibody नहीं है. Now, should I think that इसमें तो दोनों ही antigen है, then why not clumping? Clumping इसीलिए नहीं, because the amount of antibody that they have is much, or in the process of transfusion, जो भी amount of antibody that will enter is a very small quantity. 100 unit or 100, or if I talk about 100 ml of blood, 1 unit of blood, 200 ml of blood, if it is entering also this person's body, it is a small quantity of this antibody which is entering, which will not bring about effective agglutination. But equal amount of antigen antibody agar hoga, then effective agglutination hoga hi. So that's the reason when I understand, but can I say that this person who is AB, can this person actually give blood to O? Isme there is a risk. Isme there is a risk. Now why the risk this way? The risk is whenever a foreign antigen enters, the chances of production of these antibodies will be even higher. Whenever any foreign antigen enters our body, the chances of production of the antibody वैसे भी बहुत rapid हो जाता है, and the amount of antigen and antibody will be almost at par, and the process of agglutination will be much faster. तो यहाँ पे क्या हो रहा है? Antibody थोड़ा सा है, antigen इतना सारा. तो agglutination अगर थोड़ा बहुत होगा भी, हमारे बॉडी में जो normal anticoagulants जो है, that will take care. No problem. But if can I talk about the other way, the reverse way? No, the reverse way is very dangerous. So in that case, what is the method? Now again, if I talk about this type of blood entering into this, what do you think? Is it feasible? Is it advisable? AB giving blood to B. AB giving blood to B, again there's a risk. AB giving blood to B, AB giving blood to A, there's a risk. But they सकते हैं, but with proper amount of anticoagulants given, but normally there's a risk. So why there's a risk actually? Because these foreign antigens are entering the body. Can this happen? Can this happen? Can a person with A give blood to B? No, because then this will come together. Agglutination होने का बहुत ज़्यादा chances है. Can I say the other way? Can this happen? The same concept. Because these two, if they come together, the process of agglutination. So what is the theory we are understanding is, if antibody B and antigen B come together in equal proportion, there are chances of clotting. So when this is coming here, or when this is going there, this is a foreign antigen which will trigger the formation of this antibody. This is a foreign antigen which will trigger the formation of the antibody, and as it is, the proportion of, or the chances of clotting becomes much more. So in that case, it is understood that these individuals or individuals who are having blood group O, what is not there? Antigen unme nahi hai. So if this type of people take blood from others, it is generally not advisable because they don't have antigens, and other antigens entering their body trigger the formation of antibodies. So what they can majorly do is give blood. They can give blood because they don't have any antigens, so they have only antibody. That's the reason they are known as universal donors. Why are people with blood group O known as universal donors? Because they don't have antigen A and B in their blood. They don't have antigen A and B in their blood, so even if they are given, or even if their blood is getting mixed with any other types of blood, there is no chance that the antibody production is increased in the donor's body. This is giving blood to another individual. It is acting as a donor, so it is a successful donor because it does not have any antigen, so it will not trigger the formation of any antibody in another individual's body. What about this side? We are saying that this is a universal recipient. This is a universal recipient. Say suppose I understand, can this happen? What will you write as a logic? What will you state? Reason kya likhoge? No, this can be done. This can be done because the antibody A and antigen B is entering. So antigen B to already pehle se hi hai. This antigen B may trigger the formation of antibodies, but basically if this is a foreign antigen, it will trigger the formation, but it has already two types of antigens. So the process of clotting will not be that profuse and tremendous. Of course, we are saying if a person will be entering blood, giving blood, this antibody as it is comes, is there any chance that this antibody A and B, they start clumping? हो सकता है, but chances are very less because the proportion this will be small and this will be bigger amount. ये तो RBC में है ना, RBCs are in millions. These antigens are in the surface of the RBC, so this person will have this type of antigens in huge quantity in the body. तो उस comparison में जब ये antibody आएगा only in 100 ml or 200 ml, it's a much lesser proportion. तो clotting इतना profuse नहीं होगा. But again, I may say that हाँ पर ये तो है ना, this will trigger the formation of antibody, but वो भी थोड़ा सा ही होगा ना, not that much, because there will be another antigen which will not participate in the process of clotting altogether. So there are many theories here to understand, but majorly you have to come across. You get questions on this too. Why are people with AB known as recipients? Why are people with blood group O known as donors? So basically, in one or two sentences, you have to write on this concept based on the proportion of the antigen and the antibody. Antibody ज़्यादा है, antigen बहुत कम है, that may not bring about clotting. Almost equal proportion of antigen antibody है, that may generally bring about clotting. So that way. Is this all when we are talking about the blood groups? No, when we are talking about blood groups, I said on the surface of the RBC we have got the A and the B antigens. Now apart from the A and the B antigens, we also have something like the RH apart from on the surface of RBCs. So it is something like this. When I talk about the surface of the RBC, there may be something known as the RH and there may be something known as the A or the B. So on the surface of the RBCs, there are various different type of other antigens. So we say that there are specific genes which are responsible for this. Remember, for the RH, a protein produced by another gene. AB, a protein produced by certain genes actually, no? So now, if the RBC, if a person has RH antigen on the RBC surface, if a person has this Rh antigen on the RBC surface, then this person will be termed as Rh positive. A person does not have Rh, then this person will be termed as Rh negative. So what I'm talking about, I am saying that on the surface of the RBC, along with A and B antigen, there is also a third type of antigen known as the Rh antigen, rhesus. Now, this Rh antigen is something like a beta checkpoint for the process of transfusion. A and B blood group तो है ही A, B, AB, O, but apart from that, this gives us a even more better concept of transfusion, because this also may be responsible for coagulation. Okay, same concept. This is an antigen. I am saying that a person, say suppose I talk about a person A positive and I talk about a person A negative for the same type of blood, A positive and A negative. So now what's happening? Generally now, here we had understood what is the rules of transfusion. A can give blood to A, B can give blood to B. But we also studied that when we are talking about the transfusion of A, B and O, that's different. Now I'm strictly saying A can give blood to A. Is it always true? No, A positive can give blood to A positive. That is better, much better, safer. Can A positive give blood to A negative? That's risky. When the blood of A positive is entering the body of A negative, now what's happening? The same concept. Antigen is entering the body of a person who does not have any antibodies regarding this, no? So again, I'm saying that this A positive antigen, just try to understand, it's a protein. This protein is entering the body of this individual and triggering the formation of certain antibodies. तो अभी इसके बॉडी में क्या हो गया? एंटीजन भी आ गया, एंटीबॉडी भी आ गया, and there is a chance of clotting now. But इसकी बॉडी में खाली एंटीजन है, एंटीबॉडी तो नहीं है. इसकी बॉडी में एंटीबॉडी है for the rhesus antigen, but antigen तो actually नहीं है. But when I talk about the mixing of these two type of blood in the body of the donor, antigen and antibody दोनों आ गया. So now I say that this blood comes here. So yes, Rh antigen is also there and Rh antibody is also there. दोनों है. So antigen and antibody always results in the formation of a blood clump or clot. Always. Antigen and antibody reacts always to form a clumping. So that is the concept. Now if I say, you get questions this way, that say suppose I talk about a woman who conceived, who has conceived, and in this process of conception, meaning she has a fetus in her womb. In this question, you have to think this way, that I am talking about a woman and I am talking about a man. The man is Rh positive, the woman is Rh negative. Now, in the body of the child, or when the woman is conceiving, inside, in the body of the child, is there any risk when you are talking about the process of mixing of Rh positive and Rh negative blood? Because in the process of formation of the first fetus, there are chances that this individual may die out in the body of the fetus, in the body of the mother itself, in the womb of the mother itself, because now that the antigen and the antibody are Rh positive and Rh blood, negative blood coming together, again clumping may happen based on this concept. Okay, we'll continue with this again, the answering the proper concept of this. You have to understand this, that in the first pregnancy what, in the second pregnancy what, and what is the logic behind this, that why in the first pregnancy still we say that the child may survive, but not at all in the second pregnancy, the child will never survive. So that's a question that we have to complete it writing, and see you get this question as a question. Who is this? Joanna, I think I took your book.
हिंदी है ना
इसे भी बोल
इसके बाद में स्कूल का बारे क्या है ये चीज़ को पता भी है पता चला और आज तो साढ़े बाकी ये से वो पता चला क्या मेरा साढ़े 3 घंटे से मैं बाकी के दो अच्छे से लिए और फिर जो है ये लीड की जो स्टार्टिंग से है